All right, hello everyone. This is Scott Reeves, and I'm going to go through. This is FSN 329, and we're going to talk about iron. And I'm going to go about probably halfway through this lecture, probably about 12 or 13 slides. Then I think I'll stop because that's a point that I would rather go through that information in class. So, one of the things to keep in mind about iron is we're going to talk about kind of iron chemistry, and so we're going to go through an intro. We're going to talk about trace minerals overall, then the essentiality of trace minerals, and we're going to talk about iron distribution in the body, sources of iron and the RDA, talk about digestion, absorption, transport, functions, excretion, iron deficiency, and then assessment of iron status. And one of the things to keep in mind with iron is iron deficiency is a very, very common nutritional deficiencies. As a matter of fact, the World Health Organization considers iron deficiency the #1 nutritional disorder in the world. It estimates as many as 80% of the world's population may be iron deficient. 80% may be iron deficient, while 30% may have iron deficiency anemia. So a couple things to take from that. Extremely common nutritional deficiency. And remember, in this case, it is a nutrient that's deficiency is definitely more common in developing countries rather than developed countries in the U.S. Is iron deficiency still common in the U.S.? It is, absolutely. The other thing to take from this is there is iron deficiency, and then there's iron deficiency anemia. So a person could be iron deficient first without being anemic, and then eventually, as a matter of fact, one of the last stages of iron deficiency, when it's been significant and for a longer period of time, is the anemia, where the red blood cells are actually affected. So iron metabolism can be compromised for quite a while before it actually shows up as an anemia. And iron deficiency develops gradually and usually begins with a negative iron balance. And then when iron intake does not meet the daily need for dietary iron, that's what happens. That negative balance first depletes the storage form of iron, and then at that point the hemoglobin levels in the red blood cells can remain normal. Then eventually it can cause the hemoglobin and red blood cells to be affected. In the case of iron, a lot of times the deficiency is because of inadequate intake. So it's not like B12 where there's a problem with absorption. Commonly it's that there is not enough taken in. And another interesting part of this is our body actually adapts significantly to our iron status. For example, if our tissue iron levels are low, we actually can absorb a lot more from the diet, and vice versa. If we have high amounts of iron, we can actually decrease iron absorption efficiency. We'll talk about the mechanisms of how that works and some of the genetics involved. And with that said, remember, it's still a significant nutritional problem for iron deficiency. So this is the first mineral that we've talked about, so I wanted to show this table. And the reason we're talking about iron now is because of what we're doing in the labs. And if we look at this table, it's divided into two sections. We have the major minerals up at the top, or the macro minerals. These are goes from calcium to magnesium, and the reason these are called the major minerals is because they're present in higher amounts in our body, and we need higher amounts in our diet. So they range in our body. This is a pound. The major minerals are those present in amounts larger than 5 grams or a teaspoon in our body. This is for a typical person that is about 60 kilograms or 132 pounds. And the pound is about 454 grams, so thus only calcium and phosphorus appear in amounts larger than a pound. So the most common mineral in our body is calcium. I think we all know that. Second most common is phosphorus, and then it ranges all the way down to magnesium. So absolutely essential minerals are the major minerals. The trace minerals, the lower part of this table, and this doesn't show all of them, but this shows quite a few of them. These are those that are present in smaller amounts in our body, and we also require smaller amounts in our diet relative to the major minerals. But these are absolutely essential. Without these, we cannot function. So even though it's small amounts, it's critical that we consume adequate amounts from our diet. There are a couple ways to define trace minerals, or sometimes they're referred to as the microminerals. These are those that are present at less than 0.01% of total body weight, or the nutrients the body needs in a concentration of less than 1 part per million. Those are the trace minerals. And in general, iron is probably the trace mineral with the highest amount in our body, and we'll talk about that. Zinc is second. Hopefully we'll get to zinc and copper this quarter. And we also have manganese and iodine and selenium. So let's look at next one. Distribution of iron in the body. The human body contains approximately 2 to 4 grams of iron, and overall I'd say that's actually quite a bit. One of the questions is, where is it distributed? And this relates again to kind of a lot of things, including what happens when we're not consuming adequate amounts of it consistently. So this is percentages. If we look at this table, I think this table's good to take note of some of the details. This is the approximate distribution of iron in adult males and females. It is different between genders. This is milligrams per kilogram of body weight. So this is not just because males are larger, because this compensates for that. This is per kilogram of body weight. So percentages, if we look at this, approximately 65% of iron in the body is part of hemoglobin. And we know hemoglobin is in red blood cells. About 10% of it is in myoglobin. So if you add those up, about 75% of our body's iron is in hemoglobin and myoglobin. And we'll talk about the differences there. But hemoglobin obviously is in red blood cells. Myoglobin is called myoglobin because myo is for muscle. So myoglobin is a protein in muscle that actually has an iron atom that allows the myoglobin to bind oxygen. So it's how muscle fibers actually obtain and utilize oxygen. So it typically stays in muscle fibers. So it's more of a stationary protein. If we look at the amounts here, this is called functional iron. We have hemoglobin and myoglobin that we have. So for example, in males, the hemoglobin is 31 milligrams per kilogram body weight. And females, it's 28, so significantly less. And myoglobin, now males tend to have more muscle, so they have more myoglobin. And that's, relative, because body composition is, males tend to be more lean, so they have more muscle. So even though this is expressed per kilogram body weight, males still have more myoglobin. Heme enzymes about the same, and then non-heme enzymes, this shows females actually have a little bit more. Now the transport iron, we're going to talk about transferrin. You guys measured some transferrin levels in your blood last week in the lab. So we know that that's the iron transporter in the blood. About the same levels there. Storage iron, this is where there's a significant difference. We're going to talk about two proteins that store iron in cells, and that's ferritin and hemosiderin. For men, significantly more, 12 milligrams per kilogram body weight, whereas females, it's about a third of that, about 4. So if you add up all these sources of iron in the body, males tend to have total iron of about 50 milligrams per kilogram body weight, and then women it's about 39, so significantly less for women. One of the reasons iron is utilized in our body is because it has different valence states. It can be present as iron plus 2, that's the reduced form, that's also known as ferrous iron. And then of course we have iron plus 3, which is the oxidized form, and that's ferric iron. So you definitely want to be able to distinguish those. That reduction or oxidation of iron allows it to do things like bind and move electrons, but also it allows it to bind oxygen. So that's a really important reason why our body uses it. Plus, iron is one of the most abundant metals in our environment. So we typically can obtain it from the diet, potentially. All right, let's look at this chart. We've seen this quite a few times before. We have different potential sources of iron in our diet. And if we start up at the top, remember iron is one of those nutrients that's part of our enrichment program here in the U.S. It's commonly part of the enrichment program in other parts of the world as well. If they have enrichment, it's a good chance iron is there. Fairly well distributed. One of the things that kind of sticks out to me is dairy products are good for lots of nutrients, but iron is not one of them. So they're notoriously low in iron, but you can see the other ones. And remember, we have both plant sources through here, but also some animal products as well are good sources of iron. So we'll talk about that. Then we have some kind of unusual sources, canned clams. I'm not sure if anyone eats those. Beef liver and then parsley. Wow, a cup of raw parsley. So eat your garnish, I guess. We have over here the RDA. The RDA for women 51 years of age and older is 8 milligrams a day. The RDA for women between the ages of 19 and 50 is 18. Right? So the difference here is menopause. So post-menopause, iron requirements in women go down because of the cessation of menstruation. So that monthly blood loss during menses can be a significant change in iron requirements and iron status. For men, the RDA is, for adult men, it's 8 milligrams a day, so significantly less than women. So let's see. Let's look at this one. Sources. Sources to remember for us. We're just going to be general here. We're going to say meats, beans, green leafy vegetables are basically the three sources that I want you to remember for iron. Now, the average content of iron in the Western diet is relatively low. It's about 5 to 7 milligrams per 1,000 kcals. So when you look at this, let's say for example, looking at a lot of FSN 210 diet analyses and other analyses, especially from students through the years, a lot of women don't consume that many calories. A lot of them will consume around 2,000 calories a day. And if you look at that, that would put you at about 10 or 14 milligrams a day of iron. If you look at this, that puts us short of the RDA for most adult women. So without making a conscious effort to consume adequate amounts of iron, it's not easy. So it's pretty common for people to be consuming less iron than they need on a daily basis. So women, especially women, need to make a conscious effort. Men kind of have kind of a built-in safety net. Men tend to eat a lot more calories, but then also they don't need as much. So it's less common to be iron deficient in men than it is in women. So let's look at this. Sources of iron, two main types of sources of iron. One is heme iron. I think we've all heard of that. So let's look at the structure over here. Oops. Here's heme iron here. This is a heme molecule. Heme is made of porphyrin, which is this ring structure without iron. As soon as iron is added, it converts from porphyrin or protoporphyrin to heme. Okay. And in the synthesis of heme, one of the last steps is, so basically cells can make porphyrin, and then there's an enzyme called ferrochelatase, and you don't have to remember that. But ferrochelatase adds that iron atom. And then this heme molecule can be used by a variety of proteins. It's not just in hemoglobin. Other proteins also use heme for their enzymatic or functional activity. If it is used by hemoglobin, then this heme molecule is surrounded by a globin peptide. It's wrapped around. You can see in the middle here this little flat. Of course, this is heme right here. See the porphyrin ring structure that basically gives it a flat or planar structure. And in the middle of it is the iron atom, and that's the one that can bind oxygen. Remember, hemoglobin has 4 subunits. Each of those subunits contains globin and a heme molecule. The difference between hemoglobin and myoglobin is a single subunit, but it basically is the same. It's globin wrapped around heme. That's what myoglobin is. So when hemoglobin delivers oxygen to a muscle cell, that oxygen can diffuse into the muscle cell, and the myoglobin within that muscle cell can temporarily bind that oxygen to give it a little bit of a storage site of oxygen. Okay? So there are dietary sources of heme iron. They're derived mainly from hemoglobin and myoglobin in animal products. So the heme iron is mostly in animal products. About 50% of the iron in meat, fish, and poultry is heme iron. Heme iron in the diet has an absorption efficiency of about 25%. So when we consume heme from our diet, we absorb about 25% of it. And again, what's interesting, I would say, is that's relatively low. I would say only about 25% of it is absorbed. And a lot of us, I think, we say, oh, heme is the best source of iron in the diet. It is, but still only absorbed at about 25%. Non-heme iron, which is basically everything else, all of the other forms of iron in the diet that are not heme are called non-heme. So sources, primarily in plants, but it's still bound to different proteins. One thing to keep in mind with iron, in our body and also in some of our dietary sources, iron is always bound to proteins. In general, it's bound to proteins. And the reason is because free iron in our body can be harmful. And it can also, in order for it to function, it's typically bound to proteins. For it to be stored, it's bound to proteins because free iron can be harmful in the body. So even in the dietary sources, we want to remember iron is basically always tends to be bound to proteins. So in plants, even though it's not heme, it's not wrapped around by globin, it still is bound to other proteins. So when we absorb it, it's got to be digested from the proteins first. Usually about 90% of dietary iron comes from non-heme. So we tend to get more non-heme iron from our diet than heme iron. Non-heme iron has an absorption efficiency of about 17%, even lower than heme iron. Now because most of our dietary iron comes from non-heme, the overall absorption efficiency of iron is about 18%, but that can range from 10 to 35% depending on iron status. So when they do the DRIs, they assume people are going to absorb about 18%, even though if a person is deficient, they can absorb significantly more iron. So for example, if there is a pregnant woman that needs more iron, she can absorb up to 35%. There's an adolescent that's growing rapidly. Their absorption efficiency can be up around 35%. If a person has significant amounts of iron already in their body, absorption efficiency can go down to 10%. So it can fluctuate significantly based on iron status. Okay, let's see here. Sources. Some good sources of iron or significant sources of iron. Well, fortified foods, fortified or enriched foods, things like pasta, bread, cereals, flour, they have iron present, and it is added as non-heme iron. Okay, it'll tend to be in something like ferrous sulfate or some other salt form of iron. It's not bound to heme, and I think it's because it's less stable as heme if it's in a supplement. So they put it in as elemental iron, and One thing to keep in mind, now I said that absorption efficiency can be altered depending on iron status. That only involves non-heme iron. So the regulation of iron absorption only involves non-heme iron. It's not heme iron. So let's keep that in mind. Some enhancers of non-heme iron absorption. There are some other parts of the food that we consume that actually have an effect on how much of iron is actually absorbed. An example is ascorbic acid. So vitamin C can actually reduce iron to its +2 form, and that improves iron absorption efficiency. Fructose, I believe, and other sugars in the diet, like even lactose, can help improve iron absorption. It helps keep the iron soluble in the gut. And what that means is iron can become insoluble. It can bind to other iron atoms and actually form kind of a like an iron crystal, and that can decrease its absorption efficiency. So some types of sugars, especially fructose, can actually help improve iron absorption efficiency. There's also what are called meat, fish, poultry factors, MFP factors. These are considered to be small peptides or amino acids that help keep iron soluble in the gut and can enhance its absorption efficiency. So when you consume animal products, not only do you get heme iron, which is a good source of iron because of its efficiency of absorption, there's also digestion products from meat, fish, and poultry that help iron absorption. And I think this is explained by certain animal products have more contractile proteins like actin and myosin. When they're digested in the gut, they tend to make peptides that help bind iron and keep it absorbed, keep it soluble so it can be absorbed by the enterocytes. Then there are some inhibitors of non-heme iron absorption. Remember, we're talking about modification of the efficiency of iron absorption, so we're talking about non-heme iron. Polyphenols, which are sometimes referred to as polyhydroxyphenols, things that are in, for example, tea and coffee, can actually reduce iron absorption. For example, they say if you drink coffee and it's in the gut at the same time as iron, it can reduce iron absorption up to about 40% reduction. So the solution to that is eat more iron if you have to have your coffee. Oxalates and phytates, these are negatively charged organic molecules. They have phosphates that are negatively charged. They can actually bind that positively charged iron atom and make it unavailable for absorption, so they can decrease it sometimes. And then other divalent cations, something like calcium, it's not recommended to take calcium and iron supplements at the same time because one of the reasons is they compete for a similar transporter because they're both positively charged, and calcium is usually present in the gut at higher amounts than iron. So it will decrease iron absorption efficiency. It can. So let's go through this slide and we'll spend a little bit of time on this. And it looks bad, but we're gonna actually looks fascinating. We're gonna break this up into a couple different parts. So to orient what we're looking at here, we have a single enterocyte here, brush border membrane, basolateral membrane. We have some iron processes that are occurring inside the cell. We have some description of what's happening on this slide down below in a few points 1 through 4. We also have what's happening in the stomach and the small intestine, and then over here we have the portal vein or the portal blood. So we're going to take some of these all the way through into the portal vein. For iron absorption. So let's do animal protein first. Okay, a significant amount of that iron in animal products is iron that is bound to proteins in the form of either hemoglobin or myoglobin. So in the stomach, hydrochloric acid and proteases, specifically pepsin, separates the heme from the globin, so separates the protein apart. Then there's more proteases that are present in the small intestine. That completes the separation of heme from proteins. Remember, heme is also used by other proteins, not just globin. So those proteases can separate heme from proteins. Once you have heme in the small intestine, the heme binds to a transporter called heme carrier protein 1. That's down here, HCP1. That's a transporter that is believed to bind heme specifically, transported into the enterocyte. Once the heme gets into the enterocyte, the iron atom is removed. So if we go back to this, now this is heme, it would be in the enterocyte. What the cell does, it uses heme oxygenase to remove this iron. This iron comes out, so you're left with protoporphyrin, just the ring structure, which is no longer relevant for what we're talking about. The iron then binds a variety of ligands. And remember, in cells, iron is typically always bound to some type of ligand or protein. And you can refer to proteins as ligands as well. The iron is going to be in its reduced form at that point, so it's ferrous iron. It can bind ligands. And the ligands can be things like amino acids, small peptides, so these could be, or a protein known as mobile ferrin. And as the name implies, mobile ferrin is a small protein that transports iron through enterocytes. Okay, so this could be amino acids, small peptides, or a protein known as mobile ferrin, but the iron is going to be bound. The iron is then transported to the basolateral membrane, where it binds a protein called ferroportin. And as this describes, ferroportin transports iron out of the enterocyte as hephaestin oxidizes it so it can attach to transferrin for transport in the blood. Okay, so kind of a multiple step process here. The iron binds ferroportin. That's a plasma membrane transporter. The enzyme hephaestin is an enzyme that oxidizes iron as it's released from enterocytes. Hephaestin is a metalloenzyme. It uses a copper ion. So here's copper in its cuprous form, so it's +2, it's reduced copper. So it can actually oxidize iron. So it goes from +2 to +3. And as it does that, it converts, so I think I said this wrong. This is the oxidized form. This is actually cupric, sorry. Hephaestin is in its +2 form. So when it oxidizes iron, it takes an electron. Remember, electrons are negatively charged. So when hephaestin takes an electron from iron +2, it converts it to iron +3. The iron is now more positively charged because it lost an electron, so now it's oxidized. When that happens, the copper of hephaestin, lost my cursor, is reduced. So it picked up that electron. So hephaestin oxidizes iron so it can bind to transferrin. We said in lab last week that transferrin can only bind oxidized iron. So hephaestin is an enzyme that is present on the outside of enterocytes. As ferroportin transports iron out of the cell, hephaestin oxidizes it so the iron can then bind to transferrin. Right, so here's a transferrin protein. In this case, it's bound to 2 oxidized iron atoms. Remember, sometimes transferrin binds 0, 1, or 2 iron atoms, but it's always oxidized. Transferrin can then transport the iron from the portal vein to the liver. Okay? Now, remember, copper is an important part of this. There is another enzyme that is called ceruloplasmin. CERULOPLASMIN, as in Nancy. Ceruloplasmin. Ceruloplasmin has the same function of hephaestin, but ceruloplasmin oxidizes iron in all other tissues other than enterocytes. So ceruloplasmin does the same thing as hephaestin, but ceruloplasmin does it in all the other tissues. So when iron leaves a cell other than enterocytes, ceruloplasmin oxidizes it so it can bind transferrin. Okay? And iron absolutely has to be oxidized for it to bind transferrin. And one example of this is in copper deficiency. You previously worked in copper metabolism and everything. That's where a lot of my research was done previously. What we found is in copper deficient rats, they became extremely anemic. But we also found that copper deficient rats had really high levels of iron in their liver, extremely high levels in their liver, and yet they were extremely anemic. They would have hematocrits of sometimes 12 to 15, which is really low, and their whole body was anemic. They looked pale. They were they'd get lethargic because of it. And what we found was the iron was actually accumulating in the liver, so it couldn't be used by other tissues to make red blood cells. So the iron would sequester in the liver, couldn't get to the bone marrow to be used for red blood cell formation. The reason it was accumulating in the liver is because ceruloplasmin needs a copper ion. Copper deficient rats had compromised ceruloplasmin activity, so the iron that was actually in the liver couldn't be released from the liver because it couldn't be oxidized by ceruloplasmin. So the rats had high amounts of iron in their liver because they had copper deficient ceruloplasmin levels. Okay, so that's an important point of copper. Again, of copper and iron in relationship. So I think that takes care of the first part of this. The second part is not as bad because there's commonality as it goes through this. Now, we said that overall most of the dietary protein that we consume is actually in the non-heme form, and even though it is in the non-heme form, it's still bound to proteins overall. So as it goes through the stomach, hydrochloric acids and proteases, specifically pepsin, releases the non-heme iron from proteins, or some of it, so the food components separate. In the small intestine, there are additional enzymes that separate iron. The iron in the gut can be either in the +2 or +3 form, if it's elemental in this case. Okay, your textbook talks in some detail about this. Don't worry too much about that. Just remember, iron in our gut can either be +2 or +3. And as the iron is in the small intestine, but it's in the reduced form, it can bind to a transporter called DMT1, which is divalent mineral transporter 1. That's a transporter that binds and transports +2 iron, ferrous iron, goes into the cell. Then once DMT1 releases it into the cell, it binds to other ligands, and then the steps are similar to what we just talked about. Okay? Some of the iron in the gut is in the +3 form. It's oxidized. Some of that iron can be reduced by an enzyme called D-site B, which is duodenyl cytochrome B reductase. That enzyme, and this is a little bit, anyway, that enzyme reduces iron to the +2 form so it can bind DMT1. Okay. Some of that +3 iron is actually bound to ligands, like for example phytates and oxalates. Some of it remains bound to those ligands and it's not absorbed. But some of that +3 iron can actually be separated from the ligand and it can bind to a protein called integrin. Now integrin is a little bit still not completely characterized at this point, but they think this protein integrin can actually transport some of this oxidized iron into the cell, and then there's an intracellular reductase that can convert it from +3 to +2. Okay, so in the cell, for it to bind to these ligands of mobile ferrin, it's reduced. Then, once it's reduced, it goes through ferroportin and into the portal vein, as we talked about before. Okay, so there's a few different ways that iron gets into the cells, and one way to distinguish is whether or not it's heme or non-heme, and then if it's non-heme, whether or not it's reduced or oxidized. Okay, so that's all important details. And again, the iron's always going to be bound to a protein. There's a couple other proteins that I want you to add here. And those proteins are ferritin. No, I don't have it here. And hemosiderin. So here's the spelling of hemosiderin. And here's ferritin. So down here in this slide, in your notes, write ferritin, and then put a bidirectional arrow, hemosiderin. I have this slide here so you can kind of write out what each one of these proteins does. So far we did this one, DMT1, integrin, ferroportin, hephaestin, ferritin. Ferritin is an intracellular protein that functions to store iron. So it's an intracellular iron storage protein. That's what ferritin is. Hemosiderin is made from ferritin. So hemosiderin is also an intracellular iron storage protein. So this doesn't show it, but when iron goes into an enterocyte, some of that iron can actually be bound to ferritin. And that provides an important protective mechanism. But ferritin also provides a method of regulation of iron absorption. Iron that's bound to ferritin can sometimes stay in that enterocyte until the enterocyte is sloughed. Now, if iron is unbound in cells, it can become an oxidant and it can harm other molecules within cells. So what our body does is when more iron comes into a cell, it increases ferritin levels so the ferritin can bind the iron so it doesn't become free and doesn't become harmful. So in general, the more iron that goes into a cell, the more ferritin that cell makes. In the case of enterocytes, then that iron can go in, bind to ferritin. That enterocyte can hold on to the iron until the enterocyte is sloughed. So as our iron intake goes up and we become iron adequate, ferritin levels in the enterocytes go up. And that can help us decrease the amount of iron that actually goes through the cell and into the blood, and vice versa. If a person's iron deficient, they make smaller amounts of ferritin, So the iron will have a tendency to go into the blood rather than be bound in the enterocyte and lost in the feces when the enterocyte sloughs. Okay, so hemosiderin is just kind of related to ferritin in that level as well. So that's one of the main points of regulation of how we can actually change our iron absorption efficiency. Another point of regulation is DMT1. Now, DMT1 is a transporter, allows iron to go into the cell. So if a person is iron deficient, guess what happens to DMT1 levels? They tend to go up. If a person has adequate amounts of iron already, what happens to DMT1? Levels go down. We're going to talk about the regulation of how that works. I want you to be able to explain that to me on a quiz or an exam. We'll talk details in a second. Okay, so that's 2 points of regulation of iron absorption efficiency. Another one is at ferroportin. And as you would suspect, in general, as a person becomes more iron adequate, ferroportin activity goes down. As a person becomes iron deficient, ferroportin levels go up. One of the questions is, how does our body actually know how much iron we have, and then how is that communicated to enterocytes? That's where hepcidin comes in. So hepcidin is a hormone made by the liver, and it is a key component involved in iron absorption regulation. So hepcidin is a hormone made by the liver. When hepatic iron goes up, more hepcidin is made by the liver and released into the blood. Hepcidin travels through the blood, binds ferroportin, causes ferroportin to move from the basolateral membrane to the cytosol and get degraded. So when hepcidin binds ferroportin, triggers its release from the basolateral membrane, ferroportin goes intracellular and is degraded. So when iron levels in the liver go up, hepcidin, the hormone, is released. That decreases ferroportin activity. More iron stays in the enterocyte and is left in the feces. That's an important feedback point. And the opposite is true. When hepatic iron goes down, less hepcidin is released, ferroportin stays active. So more iron goes through ferroportin into the portal vein. Okay? There's a genetic disease, just to kind of illustrate the importance of this, there's a genetic disease where the liver's not making adequate amounts of hepcidin, and even though the tissues, including the liver, are accumulating iron, there's no downregulation because the hepcidin isn't being released in these individuals because of a genetic abnormality. I'll talk to you about that later. There's no feedback, so the person continues to absorb iron at high levels even though they have high iron, and that iron can accumulate in tissues, overwhelm the tissues, and become toxic, and eventually that can cause liver dysfunction and other abnormalities that in some people can actually become eventually lethal. That's called hemochromatosis, and we'll talk about that. Now, we'll talk about how that's treated as well. It's the most common genetic disorder involving nutrition. So, another point of this is, well, I found out a few years ago there were some studies that showed that obesity can actually increase incidence of iron deficiency. Hepcidin is actually fairly sensitive to inflammation, and if a person has significant amounts of adipose tissue and they're obese, that can actually increase their state of inflammation. That can that can increase hepcidin release. So in obesity, sometimes you find higher levels of hepcidin in the blood, and that decreases their iron absorption efficiency, so it can increase the incidence of anemia or iron deficiency in those individuals. Okay, so pretty interesting about how our body can actually sense levels of iron and actually affect iron absorption efficiency. All right, let's look at, I think we defined everything here. Kind of an overview of what we talked about. It's a little bit different way of looking at it. This is iron that's ingested in the food. Iron will go into the cells with a variety of mechanisms. It can bind ferritin, and this shows the iron being released. This is kind of a weird diagram. Shows the ferritin actually, some of the iron can be released from the ferritin, go through ferroportin, be oxidized by hephaestin and bind transferrin. But sometimes that iron actually doesn't go through, stays in the cell. Remember, it can be sloughed. So sometimes they're showing the iron can actually, some of it bypasses ferritin, goes directly into the blood, binds transferrin. Transferrin moves it around. So basically that iron can be incorporated as myoglobin in muscle cells. A lot of that iron is going to be taken up by bone marrow, be used to make red blood cells. Red blood cells, remember, stay in our blood for 60 to 90 days, and then they can be turned over. And one of the tissues that breaks down red blood cells is the liver. One of the main ones is the spleen. The other one is the liver, and that's why liver can accumulate so much iron, needs to be exported out, because as I mentioned before, that iron can then be oxidized by ceruloplasmin, bind transferrin, and be released from the liver, so it can kind of recycle through this. So our body recycles and reuses a lot of those iron atoms, and again, one of the main functions is in bone marrow to make red blood cells. Okay, it can recycle it unless we're losing that iron as blood loss. For example, blood loss leads to iron loss, such as in the menses. So that can be an issue. The liver and spleen break down red blood cells. Most iron is recycled. Okay, iron loss is GI tract cells sloughed off. That's a mucosal block, it's referred to as. Ferritin is part of at least two protective roles. Ferritin protects cells from having free iron, and ferritin also helps to regulate iron absorption from enterocytes. Okay. Now, one of the questions that we're going to address is, if cells need iron, how do they actually get iron from transferrin? And we'll see that cells that need iron can actually make transferrin receptors, and that's how they grab transferrin from the blood and incorporate its iron. We'll see that's an important point of regulation. A little bit more about storage of iron and ferritin. Ferritin is a protein that can store large amounts of iron. It has 24 subunits and stores up to 4,500 iron atoms. So here's a little depiction of ferritin. Each one of these little kind of rod structures is a subunit of ferritin. You can see these little pores or openings, and the iron can actually go in there. And iron is oxidized as it enters ferritin, so it'll go in here. It's actually oxidized. That allows it to form what are called ferric hydroxide crystals, or it forms this kind of crystal structure here. And it can accumulate significant amounts of iron. Remember, this is in the cytosol of a lot of different cells, and the ferritin is accumulating the iron here, so it's not free in the cell. And this doesn't show it specifically, but iron can come in and be oxidized, or iron can actually be released in kind of a controlled rate, and as it's released, it's reduced and then it's bound to other proteins in the cell. So it can kind of go both ways through this. In our body, there's three main sites for iron storage. One is the liver. Liver stores quite a bit of iron. The other one is bone marrow, and the other one is the spleen. Those are probably the three tissues that are most important for iron storage in the body. Here's transferrin, and this shows transferrin is actually a dimer, and it has, for example, here's the N-terminus of peptide number 1, N-terminus of peptide number 2, so this kind of shows the ribbon structure. So we'd start with N1 here, and if we were able to follow this around, I can't really follow this. I guess it's here. Goes to the C1 of this. These little round circles are iron atoms. So transferrin has 2 iron binding sites. This transferrin molecule actually has 2 iron atoms. But as we mentioned before, it can either store 2 or 1 or 0. And this is the main iron transporter in the blood. Transferrin binds to transferrin receptors on cells and is then endocytosed. So transferrin receptors are found on nearly all cells. Those transferrin receptors are regulated based on the iron status of a cell. So transferrin is synthesized in the liver, travels through the blood, picks up iron when it's oxidized, and then if a cell has low iron, that cell can make more transferrin receptors. If a cell has high iron already, that cell will make fewer transferrin receptors, so it won't absorb it into the cell. This is probably one more slide, then we'll quit. Here we have a cell, and again, this is almost any cell type in the body, and this shows how the cell actually takes in iron bound to transferrin and then incorporates it in its cellular components. So we have the blood, transferrin molecules in the blood. This receptor actually binds transferrin. So this is remember transferrin typically is a dimer. Each one of these transferrin molecules is bound to an iron. Remember, it's oxidized. The receptor then binds the transferrin. It's endocytosed. So this complex is endocytosed. The transferrin receptor can be recycled back to the cell surface. Some of it actually can end up in the serum. Transferrin ends up in this endosome with its receptor. The cell drops the pH excuse me, of the endosome and similar to the LDL receptor process, cell drops the pH of the endosome, transferrin receptor, transferrin separate. The iron separates from transferrin. The iron can be then transported out of the endosome, reduced, bound to a ligand in the cell. The cell can use the iron if it needs to for metabolic reactions, or the iron can be oxidized and stored in ferritin. And here's the ferric hydroxide crystals I mentioned. And remember, ferritin can go back and forth between hemosiderin. It's believed it can be just kind of modified enzymatically. Same thing, hemosiderin stores iron as well. That iron, though, can be released, can be reduced, released, and then be used in the cell. Typically, the functional form of iron is initially the reduced form. Okay? So if a cell needs more iron, it makes more receptors. If a cell needs to decrease its iron, it makes fewer receptors. We'll talk about how all of that is regulated coming up. Okay? So here's the next concept we're going to talk about for iron regulation, and I think this is probably better to talk about in class because this is a new mechanism that is probably, I can draw some things on the board that will hopefully explain this better. So I'm going to stop there, and that'll conclude this YouTube, and then we'll pick up from here when we get in class.